Hi, I'm Simon Livo. I'm an evolutionary ecologist at the University of New Orleans.
Hi, I'm Jerry Husak. I'm an evolutionary physiologist at the University of St. Thomas.
Welcome to Biology Exit Interviews, where we sit down with biologists who've had long and distinguished careers to talk about their science and about how biology has changed over the course of their working lives.
And whether you're brand new to integrative biology or you've been doing it for decades, I hope you'll join us and find something interesting in our stories. Thanks for joining us.
Welcome to Biology Exit Interviews. Today, Jerry and I are sitting down talking to Diana Hews. Diana is a professor emeritus in the Department of Biology at Indiana State University. She has tons of publications. She's had a very long and prolific career. She's a fellow of the Animal Behavior Society, editor of a bunch of journals. She's worked with a variety of species over the course of her career, and her work on hormonal mechanisms of behavior and signal expression in lizards has been extremely influential and changed the way that we think about animal signaling. So, welcome, Diane. Thanks for coming.
Thank you.
So Diana, just to sort of clue you in, the reason we're doing this, it all stems from a meeting that Jerry and I had with David Wake many years ago at a conference, where he told us all about his the beginnings of his career, which was, having a job lined up before he graduated, having an NSF grant before he started at that job, and in many ways, he was describing, a world of academia that is unrecognizable to most of us today. So maybe we can start there. If you can tell us a little bit about your pathway into science, what got you interested in science, and how your career got started.
I got into science my undergraduate years because I was an outdoors type person, and I thought I want to do something that lets me be outdoors. So I decided I would be a biology major as an undergrad. Once in college, I got, a taste of some research. I went to a small liberal arts college that didn't have a lot of research going on, but I got a taste of it. And got encouraged to go to graduate school. I went to Oregon State University for a master's, and I was greatly influenced there by my peer graduate students. James Lukchenko and Bruce Mengi were there, who are sort of world-class rocky intertidal ecologists. Not anything I had to do with, but I was interested in ecology and animal behavior. And so being around those students really influenced me. And I think that's the case throughout my whole career, really, being around your colleagues. And then I went on to graduate school, had some ideas about how to write a short-term fellowship at the Smithsonian Tropical Research Institute, which I learned about from the Lukchenko Mengi students. And that's how I got introduced to the person who ended up being my dissertation advisor, Mike Ryan. I went down to where he was doing his postdoc, Berkeley, to learn something about the frogs that I was thinking of writing my short-term fellowship on between my master's and my PhD. And then through talking with him, I realized he was starting up a job at UT Austin, and I should apply to work with him as a doctoral student. And I applied to several other places, but I ended up going there. And I'm very glad I did. It greatly influenced how I thought about biology, frankly. It was a great mix of faculty there when I got there.
Yeah, that's
And
interesting. Interesting that you mentioned Mike because we've actually interviewed him as well, and he had lots of fun stories to tell about
oh yeah,
working at
a
Spry.
storyteller, sure. But I went there and there were all these very young professors there. I mean, David Hillis was there, Mark Kirkpatrick, Craig Pease. These are all people that are population geneticists or ecologists, evolutionary biologists. And we would go to the bar, taverns, drink beer, and shoot the bull all the time. So it was just very, very exciting time to be there, frankly. Yeah. And I wrote a gazillion grants to do my PhD. Somehow I had the wherewithal to go. Mike Ryan, of course, worked primarily on frogs, although I did a little bit of fish research with him too. But I decided I did not want to work on frogs because that's what Mike worked on. So I worked on sexual selection, but went in a lizard direction because I had heard of this interesting population that I wrapped some ideas around for my PhD. And I think in the long run, that has really helped me, at least initially in my career, because I know when I remember students, I'm like, so and so is a student of this professor. And until that person, so and so, has published enough papers that their name rises in my brain, I remember them as Mike Ryan's graduate student, and not Diana Hews. And I saw several of Mike's other students sort of go through that period where they had been working on frogs for a long time, and their name never really got a lot of name recognition. And I think was, oh, that's just another Mike Ryan student. So I thought for some reason I went that way. I'm not quite sure why, but I think it was important. And then I wrote some postdoctoral. I wrote a National Institutes of Health, NRSA, National Research Service Award, which is it was an individual grant, not an institutional grant. And, so there I am in the last year of my PhD, trying to finish up my PhD and writing the postdoctoral grant, which I think many of us went through, struggling to find the postdoc. Most of the time, postdocs is what you needed to do to move on in your career. And so I was lucky enough to get that, and that kind of got me into hormones, which ended up in my mind being a second PhD. I was able to be there for 4 years, 1 year supported by Arizona State University on one of their fellowships, and then 3 years on my NIH NRSA. And it really did, I didn't know how to hold a pipette. So it was completely retooling for me. And that's Mike Ryan, he's this evolutionary ecological researcher and knows a lot about mechanisms. And so that was the big influence of Mike Ryan, was to get me interested in mechanisms and hormones.
There's been a lot of changes to the way that people think about mechanisms, over the last couple of decades. Do you feel like we're any better at integrating mechanisms into the stuff that we do now, or do you think that it's still very siloed? Because we always talk about integrative biology and how important it is, but sometimes it feels like there's still a bit of a separation.
Yeah, it's well, so within my discipline, mechanisms. Well, you guys are behavior too. In mechanisms of behavior, we're now getting into, neurogenomics, what genes are being expressed, what genes are being, epigenetics of the neurogenomic landscape. And so there are a handful of people that are doing that. I think the whole next generation is doing that, but we're still just scratching the surface. It's like, in my mind, I think it's the beginning of when we started to be able to sequence genes. It's like, yeah, we have these 400 genes that are being turned on and these 300 that are being turned off, and they're in these 8 different kinds of very complicated, networks that we can identify. And so it's still at the beginning of that next step. And so I don't know where, I think it will ultimately go somewhere, but it's very complicated. And I think the additional complication I ran up against this when I wrote NIH grants, and to some extent NSF grants. I work on lizards, so that's a non-traditional system. And if you get reviewed by mice researchers, they're like, well, why are you doing this old-fashioned blah, blah, blah? And it's like, because that's what we need to do to start in a non-traditional system. We have to build the story, right? And so that, even at National Science Foundation, that was not appreciated comparative research was not appreciated in some of the other panels. And I always felt like I was caught between, the animal behavior panel and then the neuroscience panel. And they were 2 different audiences. You didn't know who your grant was going to go to. And so half the time you got 4 people that liked it and 4 people that hated it. So that was kind of challenging. And I think it's evolved a bit in NSF so that you have people that maybe don't understand the methods, but appreciate the methods. the neurogenomics. I barely understand it, but I can appreciate them.
Yeah, so thinking about that kind of shift from kind of one technique to another, how do you feel in your kind of specific area, your discipline? Has that gone? Like, do you feel like people move on when it's time to move on, or do they move on when something new and shiny comes along? Um, at the expense of previous techniques that had been successful maybe for many years, but now here's something new that funding agencies want to see. So maybe we just go with that instead of either using previous methods or only partially using those. Do you feel like people move along at a good pace there with what they use, or do they kind of just follow trends, or what would you say you've seen?
Well, I think I've seen a little of everything. People always follow trends, unfortunately. I mean, it's just like science has always been like that. You publish something once and then it's not new anymore. It'd really be nice if we showed this 10 or 11 or 12 times in different species. Um, I mean, even hormones. I've had people cite my paper in some European lizard, and they never validate what the delivery level of the implant was. There's all these errors in what they do. And that happened in the 90s. So there's lots of examples of people jumping on a bandwagon, not completely understanding it. And some of these things get published. It depends on who the reviewer is. And so that's frustrating to see shoddy science being published. But we see that every generation, right? We always have that. And it's
Avoid it,
sort
I
of
guess.
yeah, I don't think it'll go away, unfortunately. Shoddy statistics. So as far as so what I'm calling neurogenomics and gene expression, there's a couple of labs that really are moving forward. I think Kim Rosvo's lab is doing an outstanding job, and her postdocs and where they're continuing on in starting to really get at. They've got very much more specific questions because they've done the legwork to document the string of effects that hormone level affecting androgen receptor levels, affecting where they are in the brain, so they know the system well. So there are labs that aren't just bandwagging it. They're really working it and getting more and more depth of understanding. But certainly lots of bandwagging too. I mean, we have a paper out, my collaborator, Amelia Martins, and I, and some other people at Arizona State University on transcriptomics following a behavioral encounter in lizards where we had control versus not control chemical signals and get a whole bunch of genes being expressed, and we can do the gene ontology and say, well, but we have no idea really what's going on. But you have to start somewhere, right? So it's a bit of bandwagon. It's a bit of you have to start somewhere.
So you mentioned that you switched your lizards so you could sort of move out of Mike Ryan's shadow. But when you look at sort of the scientific landscape now, the opportunity probably looks a little bit different, right? So I think one of the issues that a lot of beginning graduate students face is like, well, it seems like everything's been done. Like, well, what do I do now? So if you were starting off today, or what advice would you give to a young graduate student about where you think there's a lot of opportunity?
Independent of funding or
Well,
not?
let's go with that. Just
Okay.
scientifically.
Yeah. I mean, so intellectually, there are so many things we don't know, and it's so easy to forget what we don't know when you read the literature and how people write their papers. Do we truly know A, B, C, D, E happens, the sequence? There's a lot of jumping over some of the intermediate steps. And so it gets nitty gritty. But again, assuming you're sort of interested in mechanisms, connecting the dots is really important. Now, the way to really, I think, move the discipline forward, though, is as always, find a really good system that's good for asking the question that you're interested in. And that takes reading and knowledge of different systems, which I think is hard when you're a beginning grad student. That's actually why I'm a big fan of master's theses, because it just gives you a chance to develop your ability to think more critically. about what we know, what we don't know, and a given research paper that you read and how strong it is or isn't. So I also, going to conferences, that's where you get so excited about you see what other people are doing. It raises a zillion questions in your head. So being able to talk science with your colleagues, people, your other students and faculty, especially conferences like Society for Integrative and Comparative Bio, people are very, very open to talking to people and encouraging graduate students. So you kind of see the landscape there and see the huge array of questions that are possible to be addressed. And I think that is helpful. Of course, it's hard to go to a conference your first year when you don't have any data and whether you can have funding available to do that. But even smaller conferences can be useful in that way.
Yeah, that's true. Yeah, I'm gonna go back to you mentioned masters just now, and you mentioned it earlier too when you said that you've had a master's. I did the same. I went into a master's program and then did PhD after. I think Simon, you did the same, if I recall correctly.
Yeah, I also got a master's degree first.
And it seems like I'm, at least in my sphere of where I see students heading, is that's kind of gone away, or at least the push for graduate work has largely gone to finish your undergraduate degree and then jump straight to a PhD program. And I'm curious if you have seen that also and what you think that kind of means in terms of, this how quickly science is done and just how it's done with those rapid movements through school.
Yeah, I haven't seen that push here. I think it's a idiosyncratic to the advisor and the professor. Our department doesn't necessarily have, at least it didn't have that push. I think, it's hard for students they want to get in and get out and get done, right? And so for them to go a master's, 9 times out of 10, you have a student who has a little bit of insecurity and doesn't really know what they want to do, and so to really push on that and say, look, here's a little 2-year comfort window you have of showing that you can write, that you can complete something, and you get to develop your ideas. It's a really great idea, so I push it really hard. And the old mentality, like when Dave Wake was around and other people, where master's degree were the terminal exit degree, that's from the 1960s. And there's certainly not that attitude at most universities. Maybe there is still at Berkeley or something. I don't know. But, the funding, frankly, our university is going through a financial crisis, funding crisis. And they probably, it's much easier to guarantee funding for master's students than it is for a doctoral student. That's dependent on how many research grants are in the department, the mechanisms of funding and so on. We here have not been pushing it one way or the other than the individual advisor.
I think one of the other things that a lot of universities have also moved towards is master's as a coursework degree as opposed to as a research degree.
Oh yeah.
But probably mostly for financial, yeah. Mostly for my institution has done it as well. I think it's really common. I don't really have a question. That's just an observation.
Yeah, and
I think it's kind of a shame in a lot of ways. See what research is all about, if it's for you.
Yeah, yeah. And so and we're getting online masters, and that's the wave of the future, right? Because it's financially attractive to universities to do that. But typically I'm taught, when I'm encouraging, it's some student who's doing research in my laboratory, doing research in someone's lab, an undergrad, and so they've gotten the research bug. And so I definitely steer them away from a non-thesis master's if I can. And, typically a thesis master's, you get a teaching stipend. Non-thesis, you pay your own way. So that's the other main benefit. And most students don't really understand that until you start talking to them about it.
So, do you want to ask about the blood extraction, Jerry?
Oh yeah. Yeah. It's kind of funny. We discovered this at some point along the way that Simon and I had both been taught by you
No kidding.
how to take blood samples from the post-orbital sinus in lizards, but it was via email. So I don't remember
Oh
where it
my
came up,
God.
but it's
And
like, oh, you too? And we were curious, like, how many times does that happen? How many people have you taught how to do that? And how many times was it via email?
oh my God. Well, I mean, I just recently did that to another new doctoral professor who was grad student of one of my collaborators, a grad student of Elia Martins. And she's done neuroscience work in frogs with Walt Wazinski and is trying to go back to lizards now. And she had never done that. So I did it over the phone with her. I think I was like holding my hands up and saying, pretend this is the lizard's eye and you want, how you want to go in this way or that way. um, and I totally forgot that I did that for Simon. So I've no doubt forgotten other people. Um, Similarly, I had a Marius Acker, one of my great graduate students, highly productive, did a study on corticosterone levels and anti-predator behavior in lizards. We never did any lizard surgeries for her PhD, so she never got surgery experience. So I actually took lizard carcasses out of the freezer and filmed myself doing surgery and suturing and tying the knots and everything and sent her that video.
I've often felt like I should apologize to you for that because I wish I still had my emails from when I was a graduate student. But I mean, in retrospect, explaining to someone over email how to do it, like you were extremely patient. So thank you so much.
Well, good.
And it's amazing it worked. Like
I
a
know.
perfect description. I was like, yeah, that's how you do it. It's that's great.
Well, I'm glad. I think it must depend. A Nolus orbit is a lot smaller, isn't it? I've always thought maybe of taking they make all these different sizes of microcapillary tubes and then heparinizing them and using the really small ones after you have done the heparinizing yourself. But never had to do that.
Avoid
I have a whole directory of files of advice, like how to mail living lizards, what to look out for and not look out for if you're mailing from Arizona, how to store your blood samples, how to collect your blood samples in the field. I have like a whole library of advice files.
We need to make that
So,
publicly available somehow. That'd be great.
well, you come up with the idea, I'll send you the files.
Well, so you talked earlier about your time at the Smithsonian Tropical Research Institute, and you've since done a lot of international field work, as well as stuff in the U.S. Do you feel like that's changed at all at any level, being able to do field work, or having students go along to do field work? Like, how has that landscape changed, do you think?
I think it's a lot easier because of email and the internet. You can figure things out much more easily, where are hotels in this town. Now, I will say, any of the international work I've done, I've always had an in-country collaborator. And so they do more of the logistics, although I was sort of a potential trip in Croatia, and I was able to, once having gone there and collaborated with people. And so being able to go on the internet, being able to do permits on the internet and email and find out what you have to do, um, that's a lot easier. But again, any serious field work that was longer than a day or two, just sort of scoping something out as a pilot, I always had an in-country collaborator. But it's easier to find those in-country collaborators now, right? Because all the publications are available on the internet. Contacting people is a lot easier, and so on.
Yeah, you don't have to go to the in-person office as much anymore, I would imagine, to get those permits.
Yeah.
Yeah.
Although I don't know my Mexican colleague, Jaime Zuniga Vega. He lives in Mexico City. He's at UNAM. And he probably did have to go to some in-person offices, but he luckily took care of all of that. And he was also very good. Mexico has some safer places and not so safer places. And when we were doing a multi-species phylogenetic study, Amelia Martins and I had certain clades that we potentially wanted to look at and species within those clades. And he went, no, you don't want to go where this species is. So we had to pick a different clade. For our questions, it worked out all right. So knowing literally the landscape and the safety of the different areas was very important for a country like Mexico.
Yeah, that would be frustrating knowing that, you get this great question, but you can't get there because you literally can't get there.
Yeah,
Or
yeah.
you can, but you won't come back.
And, when I started collaborating with people in Croatia, it began with an initiative of my university wanted to establish some potential undergrad student opportunities at University of Zagreb because one department had already done that and they went, hey, this is great. And so the dean actually sort of pointed at people in different departments and said, hey, you want to come? And I went, sure, I'll go to Croatia. And I then went online and I mean, I looked if there was anyone there at all that was someone I could collaborate with, and then went, yes, I'll go. And we just started conversations and we cooked up a 2-week project that is resulting in 2 papers. One of them's out and one of them submitted. And they've since approached me for other collaborations. I've said no and gave them some suggestions of other people because I'm retired now. But I said, I'll just come visit you on my own money.
Are there any kind of questions that you asked earlier in your career that were less acceptable at the time than they are now, or alternatively anything that you could ask back in the day that most people wouldn't be pursuing now for whatever reason?
Well, let's see. One of the questions that I initially wanted to ask for my PhD work, which was on a lizard that shows resource defense polygamy, was to measure the intensity of selection. And I was able to do that by using observed matings as my measure of fitness. And, nowadays you have to use genetic methods or you probably wouldn't even be able to get it published to determine paternity. That would be kind of hard to do where I was because of the logistics of getting it was on an island in the Gulf of California between Baja and mainland. You know, taking liquid nitrogen or taking ice out there, not really possible. Maybe liquid nitrogen, but we have to get a boat to come out to get us every 2 weeks and bring us food. So it's a deserted island. So logistics would still play a role in what kinds of questions you can ask. Intellectually, working on a non-traditional species like lizards, we were still establishing basic information. What are their circulating hormone levels? What is the behavior? What is the breeding season? Just very basic population biology and simple hormonal behavior relationships. I mean, I'm working on a paper on data that I gathered 30 years ago, I think, on orange gravid coloration in females. And I don't have hormone levels, but I do have actual ovarian state. No one has those data hardly showing through the ovarian cycle, not individuals, but through the season, how color changes and correlating that with actual ovarian condition. And I had them because I happened to be able to collect them and do dissections. It's very hard to palpate lizards and accurately determine early vitellogenic, mid vitellogenic, late vitellogenic. I don't believe anyone who does that. I can't do it, and I've tried it for 30 years because you need to validate it with dissections, and one hardly ever does that anymore because we don't like to kill her. It's not really that beneficial in general to sacrifice to kill your animals. So there's still lots of basic questions like that that we still really don't know. And people just assume, well, we've shown it in these two species, so it's probably like that in this species. Well, no, there's a lot of species variation out there. So I still think, unfortunately, the discipline is making assumptions about how biological assumptions that haven't really been validated well enough. Now, does that matter? It might not matter for a lot of questions. How many people are interested in the hormonal basis of color change? Well, you might be interested in it if you're then connecting it up to some other evolutionary theory about the costs of color change or the timing of it relative to your immune function. Then you really need to know those mechanisms. And you can't just assume that, in species one, that they're working the same way in species two. So I still think we need to do some of the same research that we've been doing for a while, but I guess it gets motivated more by not just knowing it, but knowing it so that we can ask these another level of question.
So what do you think the balance should be between, spending a lot of time and money working on a model system versus expanding those questions to other things that maybe get much less attention?
Well, we have to have both. The model systems help drive questions in these other species. But 9 times out of 10, when we find something really different and really interesting and really exciting, it's not where you were expecting to look. I still think a classic example of this that I give in my elevator, I like to say my ski lift talk, because that takes, 5 minutes, is Frank Moore at Oregon State University and Miles Orchinik, when they figured out that steroid hormone receptors for corticosterone can be on the cell membrane in addition to being inside the cell. And the reason they even asked that question is because Frank Moore had been spending 20 years understanding the role of cort in clasping behavior in the rough-skinned newt. And he knew it was a rapid cort effect. So this has to be a cell membrane receptor. So he did all that detailed work that then motivated the question. But it was a very unusual species, rough-skinned newt. And so I think a lot of times the mantra that we all say, we don't know where our basic research is going to take us. So I think we need both that basic research and then the other people that maybe are going into great depth in one species.
So you've talked a little bit already about your mentor, Mike Ryan. Are there any other people who came before you that when you were a graduate student, you were really influenced by?
Well, I think I mentioned my fellow graduate students. Again, at Oregon State University, when I was I worked with Andy Blaustein on a rodent project that failed the first 2 years, and I had to switch to tadpoles. I had a population crash, but it was my fellow grad students that really got me excited about statistics and how to ask questions and just going on and doing more research. I didn't really know what I wanted to do other than I want to do something. And so I said, okay, I'll go to grad school, get a master's. And it was my fellow grad students and journal club, kicking around papers is, I think, what really influenced it helped me develop my thinking and develop being excited about science. My postdoc mentor was very important too, Mike Moore, and he was great about really. I have a quote of his that I used all the time with my own students. I don't know. That's another question. um, but no, he was very great about sort of critical thinking and pulling apart what the possible mechanisms could be and things like that.
Well, you've had a very prolific and successful career doing research in lots of different areas with lots of different species. So what's kept you going, do you think, all that time? What is it that's really made you keep doing the research and enjoy it?
Um, I love asking questions and, the excitement of when you finally get to your statistical analysis and you look at it and you find out what's there. Um, and I love the process of actually, I like the process of writing. I've developed that, and actually, over towards the end of my career, I sort of became a self-professed science writing expert, and actually gave some talks on it and developed some tools that I started to use in my undergraduate and graduate courses, and spoke to people at conferences, which I hadn't done before. Um, so and then the one or two times a year you'll get some great undergraduate student that is really excited about what they're learning and doing, and that's always fun, right? To turn someone on to doing science. But I think my is just I like the process of doing science, of thinking about how can I ask this question? What are the what are the pluses and minuses? What can I ignore or put in the background as unexplained error? And how to then go about addressing some of those questions.
I had to laugh when you mentioned you're excited about statistical analysis because I've taught statistics to graduate students for many years, and that has not been my experience that they are excited by statistical analysis.
No, and here I am struggling to learn R at the end of my career so that I can keep that's what you have to use now practically, and it's so powerful. Um, and I won't say that I'm a statistical guru, but we don't have a lot of we only have one really good statistician in our department. And so I sort of had to help pull my own weight. Um, and it's just, I still remember as an undergrad how I got interested in ecology. And I was in an ecology class, and we went out in a lab and did a nearest neighbor thing, run a tape measure down a field and look at two different weed species and analyze nearest neighbor distance. And I was like, oh my God, what you can say with numbers. It just was so powerful to me. I still remember that. These two species were overdispersed.
Yeah, and R has really changed the landscape of how people even are doing things now because with all the specialized packages you can get to run these really sophisticated analyses has just kind of it's shaped the writing too, I would think, in a lot of
Yeah,
applications.
my last three doctoral students were all in the R world, and I, was not R trained, and I would constantly say, well, what's your model statement? And, they kind of understood what they were doing, but not completely. They still weren't completely. I'm like, where's your ANOVA table? Where are your degrees of freedom? And all of this. And so, they were using this command and not that command, so they weren't getting the ANOVA table. And just trying to talk to them about what their results were was I had to sort of learn how to read our output before I ever learned how to do R. Um, and I'm certainly not able to do any of these super complicated. That's when I go to my colleague. But yeah, it's the generalized linear models is quite exciting. What you can now analyze that we couldn't really do with simple ANOVA.
Yeah, it's astonishing how many of the analyses I do lately are some kind of mixed model. They're just so flexible.
Yeah, it took me forever to learn what's a mixed model.
And you almost never saw those in our field, just what, maybe
Mm.
10 years ago, it seems like
Yeah,
it's kind of once it kind of hit and took a hold, it just now it's everywhere, which is a good thing. They're really great
yeah.
techniques to work with.
Well, and I have a younger collaborator who worked on the bat research with me, Joy O'Keefe. So she and I were co-PIs on this contract, and then we had grad students that were funded on that. And so she's very much in tune with R, And so we always sort of had a little bit of a battle because to me, I'm the uneducated reader. And so depending on where it's going to get published, I think you need to explain, what's the reference group or whatever. You need to have at least some sentences in there that help the uneducated reader a little bit. And I think some people agree with that, some people don't. And, clearly uneducated readers are going to be out there for a while. So I think you should help them. I don't think it's going to get your paper rejected.
Hmm.
So put it in there.
Well, one thing that has changed over the last couple of decades is there was, at least for a minute there was this idea out there that scientists need to be better storytellers. And I'm not talking about popular science. I'm talking about within our own papers, that we should be using like narrative techniques within our scientific papers. And some people agree with that, and some people don't. What are your feelings on it since you're?
Um, I certainly think that there's a lot of incredibly poorly written papers out there that it's hard to figure out what is the story. So I know a little bit about I think what you're meaning by narrative techniques, but um, and I think, yes, you need to do that. I think in some disciplines, I think ecology and evolutionary biology, I'm on this side of the fence, but I think we're better at writing those kind of papers compared to cellular molecular biologists. Some of it is language. There's so much terminology in cell molecular biology. We have terminology and people think they know what it means. They don't necessarily know what it means. But yeah, definitely, I think you do need to be more sympathetic to that narrative approach, because the bottom line is if people aren't reading your paper and getting your paper, then your science is not as valuable as it could be. The whole purpose is to have it out there and influence other researchers. And so if you're not doing that influencing, then that's a problem. So I lean more towards the concept of narrative, although I don't know explicitly what some of those features might be.
Yeah, there have been some people out there who suggest that, well, we need to use storytelling techniques and treat science as a story. So not necessarily just, the language itself and clarity, but like, and my position on it has always been sort of, well, science isn't a story. Stories have beginnings, middle, and ends, and science
Yeah,
doesn't work that way. And when you try and package up something like that very neatly, it's very misleading, because one of the most exciting things about science is that often there isn't an ending. You don't know where it's going. But yeah,
yeah.
that's what I was referring to.
Well, somewhat related to this, one of my pet peeves as an editor is when someone has a variable like the time the female spent in the third of the terrarium towards this stimulus versus the other third, and they call that variable female choice. That to me is an example, it's one very narrow, but it's an example of storytelling, and it tricks the reader into thinking in a certain direction. And I always say, call the variable what it is. In the discussion, you can start talking about female choice, but the variable is time spent. And it really makes thinking about the results more complicated and difficult to put a label on it like that. It may be shorter, but it's much more complicated. And I think it's a example of advertising that I don't like, to use that kind of terminology.
I agree with you, and I think that sort of practice of just putting labels on things is responsible for a lot of semantic drift. That is one of
Ah,
my pet peeves about science. That what are we even talking about now?
yeah, yeah,
We don't understand this thing, so we put this label on it and then pretend that that corresponds to meaning. It just doesn't. It just allows
yeah,
us to.
Semantic
yeah.
drift. I like that. I'm gonna start using
Mhm.
that because you get converted. If other people label something slightly different than that, the same thing, and then you have a bunch of a label meaning
Yeah,
different things. Yeah, it's
yeah.
true.
Well, this is sort of one example, a modest example, but maternal effects. If you're genetic, so I always say non-genetic maternal effects, and people are starting to drop off non-genetics. And if you're in a hormone journal, then maybe it's clear what you're talking about. But if you're in, Proceedings of the Royal Society or something, I think you need to be very specific about what you're talking about.
I think you're right. From a quantitative genetic perspective, there are genetic maternal effects and there are non-genetic maternal effects. So you're right. I agree with you. It's important to
Yeah.
distinguish between. So one thing that we've been interested in asking people is sort of the role of contingency in your career, because sometimes we find ourselves doing things and we're not really sure how we got there, or it wasn't necessarily a deliberate choice. Are there any points in your career that are sort of influenced more by chance than by anything else?
Um, I won't see. Well, let me just give you an example of what comes to mind. It's not exactly chance, but um, so most of my lizard research was focused on testosterone. But 9 times out of 10, when we take a blood sample and we're looking at some hormone behavior relationship, we're going to measure testosterone, but we're also going to measure cort. And that's because sometimes those are differentially related to each other. So for years, I measured cort and I reported it, and I didn't really, corticosterone, stress hormone, and I didn't really, it was always secondary. It wasn't that much more work to measure it. I did it. And then after about 15 years, I was like, dang, I'm a stress physiologist. I had all these data on stress physiology. And so I started saying, I could give a talk on stress physiology. And then not too far down the road, my colleague says, why don't you join me in this bat research grant, and we can look at stress physiology in conjunction with the other population biology things that she was doing on an endangered bat species. And so it was sort of realizing that had enough under my belt to understand stress physiology. I had to review the literature, to write my papers, to talk about cort. And so I sort of accidentally became a stress physiologist. Um, and that led to the opportunity to collaborate with this colleague on the bat research that funded my last 3 grad students. So that's sort of an example of not really planning to go in that direction, but ending up being able to do it.
I'm kind of curious to hear your thoughts on kind of more broadly. So, kind of thinking over the course of your career, is there anything about academia in general, or particularly about science, that you feel is just kind of fundamentally different to you compared to when you started out at Indiana State? Is there just something that's just so different now than it used to be?
Well, what is different is probably, well, it might be idiosyncratic to an institution like Indiana State University, which is a smaller state-funded institution. Our enrollment has bounced between 14,000 and 9,000. State institutions are constantly getting their budgets cut or being overlooked. Right now, they're viewing whether certain programs should be cut if they don't graduate X number of students in Y number of years. Um, our graduate student budget has not changed in 20 years. It has the stipend, which is based on a per student stipend amount. So that's pretty scary and shocking. It makes it incredibly difficult to get doctoral students to come to your program unless you have a grant. And I think that's the case for probably many smaller state institutions. Why would anyone want to come to little old Terre Haute and Indiana State University if they could go to IU Bloomington, where they have many more peers, grad student peers, to interact with, professors, and so on and so forth? Our department has shrunk dramatically, and that may be happening a lot at other small state institutions. And so I think that's one of the most serious changes in the landscape, is the decrease in the number of colleagues for you and your graduate students to interact with. Those colleagues have grad students, so decreasing the whole peer group for your graduate students, I think in our kind of institution, is probably the biggest change. And of course, teaching loads have gone up. And so the time and the ability you have to do the kind of research has been constrained. Our university has never addressed teaching loads relative to how many grad students you're mentoring. Have you been an editor of a journal? Do you get big grants? The only time I got my teaching load reduction is when I managed to get some of the research on my NSF grant had to be done during the teaching year because of the breeding season period of the lizard. And so I got buyout then. That's the only time. So it's very challenging to try to be productive. The main reason in my later years with the decline in all these capabilities to do research is collaborating with other faculty that are at bigger universities that have more, those universities have more resources, those faculty have lower teaching loads and more time to be engaged in research. So to me, just sort of the crunch of everything that enables you to do research at a smaller institution is the big thing that I've seen changing.
Yeah,
Yeah.
the creep
Three
on
grad
time really gets, it kind of happens slowly over the years until you realize one day, wow, where did all my time go? It's now gone.
Yeah. And we can all moan and groan about different things. Our university, probably yours too, loves to do new computer system to do your, faculty activity report, new computer system to do blah, blah, blah. And then some dean comes along 5 years and changes it to yet another system, and so you're constantly spending time learning some new, Canvas or learning LMS, learning management systems. So huge amounts of time just have been offloaded onto faculty that used to be in administrative positions. My last three grad students have not gone into academia. One's in an NGO, two of them are in NGOs, and one is one of them is teaching, but she's at a non-research institution.
When you take students, graduate students these days, do they very often come in not intending to go into academia at all? Or are they, finding themselves discovering how it works and then just sort of noping out of that?
I think, yeah, one of the last three came in, had worked in biotech for a while, and he's back in biotech. And it may be I think he would like to be in academia, but realistically, because he has a family and money and stuff, he can't do it. I think some of that depends on how competitive you are and can you get a job at an institution that pays a decent salary. He also has family that doesn't want to leave Indiana, so that's complicated. And I think, the other ones come in, they're not sure. I don't think they really know. It's often, if they're coming in from an institution that they haven't really interacted with a doctoral program, graduate students as undergrads, then they don't really understand the landscape.
Given all this, do you have any advice for early career folks? Just general advice of how to be successful in today's climate, given what you've been through and what you've seen?
Yeah. I will say, if you're a woman, you're going to get put on. I have talked to many women, so this is not just my perception. You're going to be put on a lot of committees, and you need to learn to say no. I ended up being the chair of our department for 3 years, and boy, was that a lot of us have to do that in our more senior stages of our career, but just really be realistic. Get your chairperson to say in their annual review, and hopefully you have a chairperson that will do this. I am protecting my pre-tenure faculty from over too much committee work. This is the amount of committee work I expect them to do, and really try to stick to it. And it's hard, because sometimes you think it'll be interesting or neat to be on this committee or that committee, but dang, it's going to eat up your time. And then also, do not step forward with bright ideas. Your bright ideas will mean you have to do them. So even though you think and that's hard for me. I like to talk. I like, hey, let's do this, let's do this. And 9 times out of 10, you're going to be put on that subcommittee to do something, to write some long report. So anyway, I usually for a long time really kept my head down. And tried not to be noticed outside of the department. so that I didn't get put on committees. But you will get put on committees and try to say no. And I look at time and time again, you look at committees and you look at who's being the secretary, who's doing this, who's doing that. It's a woman that's doing it. Sorry, guys, but a lot of times that is the case. So really be cognizant of that and protect your time. And it's hard to say no. It's hard to say no, but you have to learn how to do it.
All right. Well, thanks again for your time. We really appreciate you chatting with us today.
So it's
Um,
been fun.
Yeah,
yeah,
thank you. That's really been really fun.